Hello, welcome to the new session of Biomolecules NCERT Revision. In this part, we will study about carbohydrates. So let us have a small introduction about the types of biomolecules. So there are two different types of biomolecules. The first one is micromolecules, and they are of small size. Micromolecules are of small size, simple chemicals that have low molecular weight. This is the basic difference, that micromolecules are made up of low molecular weight that ranges between 18 to 800 Dalton. So the biomolecule which comes under this particular range, that is 18 to 800 Dalton, that will be considered as micromolecules. And the one which are complex and are large size and usually have 1000 Dalton molecular weight, that comes under the macromolecules. So here we are going to study about the macromolecules, and in this lecture we are basically studying about carbohydrates. So the macromolecules belongs to the four classes, that is carbohydrates, proteins, lipids and nucleic acids. So these are the four different types of macromolecules. Okay. Except lipids, other macromolecules are formed by polymerization of subunits, and these are monomers. So suppose this is one monomer, this is the second monomer, this is the third monomer, this is the fourth, and all these are joined together with the help of some bonds. Then it forms a string-like structure, and it will be considered as polymer. It will be considered as polymer. And this process, by forming a chain-like structure with the help of bonds, forming some bond, this is called as polymerization. And the chain of monomer is called as a polymer. So this is the way of how the new structures, that is macromolecules, are formed by the process of polymerization of monomers. Okay, so let us start about the first macromolecules, that is carbohydrates. Carbohydrates are basically polyhydroxyaldosases or ketoses. What are ketoses? The one which contain ketone group. And what are the aldoses? The one which contain aldehyde group. So polyhydroxyaldoses or ketoses. Aldoses bear CHO group, that is aldehyde group, and some of the examples are of aldoses are glyceraldehyde, xylose, ribose, glucose and galactose. These are some of the basic aldoses which we study in some of the metabolite form, either in the case of photosynthesis process or respiration process. Ketoses, they contain CO group, and examples of some of the ketoses are dihydroxyacetone, erythrulose, ribulose, xylulose, fructose and pseudohaptulose. These are again some of the metabolites that are formed in the photosynthesis and respiration process. Carbohydrates are one of the four major class of biomolecules, along with proteins, nucleic acids and lipids. These are the four main biomolecules. They exist as both monomers and polymers. So carbohydrates exist in a form of monomer also and in the form of polymer also. Carbohydrates have a general formula CH2 whole N. So if we write, suppose this is CH2ON. Suppose the value of N is 6. Then what will be the formula? It will be written as CH2O whole 6. And when you open the bracket, it will form C6 H12O6, glucose. So this is hexose. Because here the N is 6, then it forms hexose. Carbohydrates are directly produced during the process of photosynthesis. For photosynthesis, this process is done by all the autotrophic plants, green plants. It constitutes major part of dry weight of the plants, that is about 80% cellulose. Cellulose. This is a type of a carbohydrate polymer, okay, and it constitutes about 80% of the dry weight of plant. Carbohydrates are also called as saccharides, as they are sugar as a basic component. So we know that carbohydrates are present in the form of monomer as well as polymer. So we will study about monosaccharide, that is monomer. Sugar is a simple carbohydrate monomer which cannot be hydrolyzed further because this is the simplest form, okay, and this is the smaller that cannot be converted into a more simpler form. They have a general formula CnH2nOn. So depending upon the number of carbon atom, monosaccharides are of the following types. So trioses, when they have 3 carbon, okay, we are considering the number of carbon atom in monosaccharides. So if the monosaccharide is having 3 carbon atom, it is considered as trioses. The name is trioses. An example of trioses is glyceraldehyde dihydroxyacetone. In the process of glycolysis, dihydroxyacetone 3-phosphate is formed. Phosphate group attached at the 3 carbon, third carbon. So it formed dihydroxyacetone 3-phosphate. So dihydroxyacetone is trioses. Tetroses, when the monosaccharide is having 4 carbon atom, then it is named as tetroses. An example is erythrose. Example is erythrose. Pentoses, when the monosaccharide is having 5 carbon atom, it is made up of 5 carbon atom, then it is named as pentoses. Examples are ribose and ribulose. Ribose sugar involved in RNA. Deoxyribose involved in DNA structure, formation of DNA structure. Ribulose 5-phosphate. It is very important in Calvin cycle. Hexoses, it is made up of 6 carbon atom. Examples are glucose and fructose. Haptoses, they have 7 carbon atom. When the monosaccharide is having 7 carbon, it is named as haptoses. Example, pseudohaptulose. So these are the different types of monosaccharides. Pentoses and hexoses exist in both form, that it present in open chain like structure and it also present in a ring like structure. So here you can see glucose in the form of ring like structure, and here it is present in the form of open chain. And we are having 2 types of ring forms, pyranose and furanose. So pyranose, hexagon structure with 5 carbon atom and 1 oxygen atom. So how many carbon atom is there? 1, 2, 3, 4, 5. 5 carbon atom is present. 5 carbon atom is present in the case of glucose in a ring form. This is a ring. 5 carbon atom involved in the ring and 1 oxygen. Then it forms pyranose ring structure. So glucose is having pyranose ring structure. What about furanose? Let us see. First carbon, second carbon, third carbon and 4 carbon. So this ring structure is formed by 4 carbon and 1 oxygen. Here is the 1 oxygen. So this is called as furanose ring. This is the basic difference between the glucose and fructose ring structure. Okay, so this fructose is also present in both the form, that is ring structure form as well as the open chain structure form. Glucose, most important and the common carbohydrate seen in the living world. This is one of the most common type of carbohydrate, glucose. It is the simple sugar of the blood. So sugar is present in the blood in the form of glucose, and it is the immediate source of energy. This glucose get entered inside the cell and it enter in the process of respiration. Oxygen is involved in the breakage of this glucose molecule. Okay, so that is why it is called as immediate source of energy. Glucose is stored as glycogen in the livers and muscles. When extra glucose is present in the body, it is converted into glycogen. Which organ? Liver and muscles. So in liver and muscles, extra glucose is converted into glycogen. Fructose, most common sugar in fruits. So in fruits, the sweetness is due to fructose. It is also occur in nectar and honey. It is the sweetest among all the natural sugar. So fructose is the sweetest among all the natural sugar present, and it is a reducing sugar. Derived monosaccharide. So these are the monosaccharides which are available naturally, but there are some derived monosaccharide. They get modified variously to form a number of different substances. Deoxy sugar. Deoxygenation of ribose produces deoxyribose. So this deoxyribose is present in DNA. Deoxyribose nucleic acid. So the sugar is deoxyribose. RNA, ribose nucleic acid. So here the ribose is present as a sugar in RNA structure. So deoxyribose, this is one of the modification of ribose sugar. Amino sugar. Monosaccharides have sometimes amino group attached to it. Glucosamine. Sugar acids. Ascorbic acid is a sugar acid. So when acid group is attached to any sugar, then it is called as sugar acid. So one of the example is ascorbic acid. Sugar alcohol. Glycerol is involved in the lipid synthesis. Alcohol group is attached to the monosaccharides. Okay. Sugar alcohol. Glucose. The next one is sugar alcohol. Glycerol is involved in the lipid synthesis. In this glycerol, alcohol group is present. 3 alcohol group is present and it forms glycerol. Oligosaccharides. Now bonding of one sugar to the another sugar by the covalent glycosidic bond. So if you remember, amino acids get attached to another amino acids. They form a bond, and that particular bond is called as peptide bond. That is also a covalent bond. Here, one sugar molecule is attached or form a covalent bond with another sugar molecule, and that is also a covalent bond. And what is the name of that bond? Glycosidic bond. Okay, and that forms some larger molecule which is called as oligosaccharide. So oligosaccharide contain 2 to 9 units of monosaccharide. It is not much longer structure, as we will study about the more polymer structure. Okay, so it is just 2 to 9 units of monosaccharide, then it is called as oligosaccharides. So glycosidic bond, COC linkage between first carbon of the first sugar. Glycosidic bond formed between first carbon of first sugar and hydroxyl group of another sugar. So that forms the glycosidic bond. The term oligo means few, 1, 2 to 9, and they are typically consist of, sometimes it is considered as 3 to 9 and sometimes it is 3 to 10 even. Depending upon the number of monosaccharide, oligosaccharides can be disaccharides, trisaccharides, tetrasaccharides. Okay, so there are different types of oligosaccharides also. The common disaccharides are sucrose, maltose, lactose and cellobiose. Sucrose. So we will study some of the oligosaccharides commonly found in the plants like sugarcane, beet roots and carrots. It is a storage products of photosynthesis. Plant transport carbohydrate from leaves to other organ to form in the form of sucrose. So how the glucose is transported? So glucose is first converted to oligosaccharide. Okay, oligosaccharide, this is the sucrose, and then it is transported in the plants. Sucrose has no free aldehyde or ketone group. That is why it is not a reducing sugar. The sugar is considered as reducing when they have either aldehyde group or a ketone group. So here you can see only OH group. Here all OH group are there. No CHO group or no ketone group is there. So that is why it is a non-reducing sugar. Maltose or malt sugar. It is another oligosaccharide. Maltose or malt sugar is composed of two glucose molecules. So this is also a glucose molecule and this is also a glucose molecule. Between these two glucose molecule, glycosidic bond is there. It does not occur in nature and found during the germination of starchy seeds. It produced commercially from starch by starch hydrolyzing enzyme diastase. So sometimes it is required. So it is made commercially with the help of enzyme diastase. Maltose is used in making beer. Lactose or milk sugar, another oligosaccharide. Now this is formed by the condensation of, or we can say forming a bond between one glucose and one galactose molecule. Lactose hydrolyzed by enzyme lactase present in the plasma membrane of the cells that lines the intestine. So here it get digested with the help of enzyme lactase. Many people lose this enzyme after the childhood and find that eating dietary product is discomfort. So they become lactose intolerant because this particular enzyme is not formed after the childhood. Function of small carbohydrates. So triose, glyceraldehyde and dihydroxyacetone are the important intermediates of both respiratory and photosynthetic pathway. So this is very, very important. Trioses and glyceraldehyde, dihydroxyacetone 3-phosphate. I've already told you that this is formed in glycolysis process. That is the respiratory pathway. And trioses, glyceraldehyde 3-phosphate. This is formed in photosynthetic pathway Calvin cycle. So they are some of the very important intermediates of respiratory and photosynthetic pathway. Pentose sugar, ribose, xylose form the polymers that are constitutes of hemicellulose structure. Now hemicellulose is present in some of the cell wall of the plants. So how they are formed? They are formed by some pentose sugar, ribose sugar, xylose sugar. Okay, so they all together form hemicellulose. Ribulose 1,5-bisphosphate is the acceptor of carbon dioxide in photosynthesis. So they also help in process of photosynthesis. So these are some of the small carbohydrates function. Some other are, in some plants, oligosaccharides serve as a reserve food like sucrose. Sucrose is stored in the plant's organ. Fats and amino acids are formed from the glucose and other sugar. Sometimes fats and amino acids are formed from the glucose. It is converted to amino acid, or sometimes it is converted to fats even when required. Oligosaccharides attach to cell membrane to form a coat or called glycocalyx. So here the cell envelope structure, if you remember, plasma membrane of the bacteria, then comes cell wall, and then comes the cell glycocalyx structure. So this three structure constitutively called as cell envelope. Cell envelope. So the outermost, that is called as glycocalyx. This is also made up of oligosaccharides. Ribose is an important pentose sugar which is found in the variety of chemicals like coenzyme A, FAD, NAD, NADP, and even ATP. So this is the 5-carbon structure that are present in different types of chemicals present in the plants as well as animal cell. Okay. Polysaccharides. Now we have studied about monosaccharides, oligosaccharides. Now polysaccharides. They are formed by the polymerization of large numbers of monosaccharide. So we have studied that 3 to 10, up to 3 to 10, when polymerization of monosaccharide occurs, then it is considered as oligosaccharide. But here more than this, then it will be considered as polysaccharides. They are also called as glycans. They are long chain. Right end is reduced end, whereas the left end is non-reducing end. The chain may be branched or unbranched. So they are having a long chain like structure, and that chain like structure can be branched and unbranched. We will see some of the examples that are having branch chain and unbranched chain. They are of three main types. That can be storage polysaccharides, that can be structural means of polysaccharides, and the third one is mucopolysaccharides. So polysaccharides are of three different types. So let us study the first one, that is the food storage polysaccharide. They serve as reserve food. They are two main storage polysaccharide, starch and glycogen. Now inulin is also there. That is also a type of storage polysaccharides. It is storage form of polysaccharide in plants. So starch, the first one, this is found in plants as a storage polysaccharide. It is a polyglucan homopolysaccharide. So all the monomers are of same type. They are stored in the chloroplast or the special leucoplast called amyloplast. So starch is present either in chloroplast or amyloplast. This is a special type of leucoplast. So plastids are three different types: leucoplast, chloroplast and chromoplast. Chloroplast, the one which is having green pigment, chlorophyll. Chromoplast, a part of green pigment, there are certain pigments that imparts yellow color, red color, blue color to the plants. And there are some plastids that store food material, that is called as leucoplast, and amyloplast is the one that contain starch. They occur in the form of microscopic granules called starch grains. Starch consists of two components, amylose and amylopectin. So amylose is more soluble in water than amylopectin. So amylose, you can see, it is a long chain like structure. Okay. And amylopectin, they are having a branch chain here. So 1,4-glycosidic linkage, we can see here the type of linkage present in the monomer, 1,4-glycosidic linkage. Now in amylopectin, at the site of bonding, at the site of branching, okay, alpha-1,6-glycosidic linkage is present at the point of branch. Otherwise, they are also having the alpha-1,4-glycosidic linkage only. Okay. It gives dark blue-black color with iodine. Now, here comes why the starch gives dark blue-black color when it is treated with the iodine. Because they are having a branch structure, and such a helically starch secondary structure, it can hold iodine molecule in the helical portion, and that gives the blue-black dark color. So this is the structure of starch, that they are having two components, amylose and amylopectin. Amylose is in the form of a continuous straight but the helically arranged chain where each turn contain about 6 glucose unit. So they are basically first the straight, but they form a helical structure. The successive glucose unit is linked by 1,4-alpha linkage. The amylopectin contain large number of glucose unit, and their 1,3 linkage in the case of amylopectin is seen. The next one is storage polysaccharide is glycogen. Glycogen is a polysaccharide food reserve. Now it is present in animals, bacteria and fungi. It is not present in green plants. It is popularly known as animal starch. They are popularly known as animal starch. 30,000 glucose residue and having the molecular weight of about 4.8 million. This is the residue, that is the glucose residue, number of glucose residue present in the glycogen molecule, and also the molecular weight of the glycogen molecule, 4.8 million. Glucose unit are highly branched bush-like chain. So here you can see they are branched. First they are like this. It has alpha-1,4 linkage and straight part and 1,6-alpha linkage in the branching point. So at the point of branching they are having alpha-1,6 linkage. The straight part is helically twisted with each turn having 6 glucose units. So this part is helically twisted. This part is helically twisted, and each helix contain 6 glucose unit. The third storage polysaccharide, that is inulin. It is a polymer of fructose. It is present in the roots and tubers of Dahlia. It is not metabolized when it comes in the human body, and it is totally filtered out in the kidney. So sometimes it is used for testing the kidney, whether the kidney is properly functioning or not. So inulin, this is also mentioned in NCERT. Important. Structural polysaccharide. Now they form the structural framework of the cell wall in the plants and skeleton in animals. The first one is chitin, the second most abundant organic substance. Structural polysaccharide found in the exoskeleton of some of the arthropods such as insects and crustaceans. So exoskeleton in arthropod is made up of this chitin, and it is also present in the cell wall of fungi. So cell wall of fungi is also made up of chitin. Cellulose, it is the most abundant organic substance present in the biosphere. So cellulose is also a linear polysaccharide, but it consist of repeating unit of beta glucose linked by beta 1,4 glycosidic bonds. So here we can see the structure of cellulose. These are the cellulose fiber, but they are joined. So cellulose molecule contains about 6,000 glucose residue. So there are more complex polysaccharides in nature. So they have a building block, amino sugar, and chemically modified sugar. So they are examples of such polysaccharides are glucosamine and N-acetylglucosamine. So if you remember, glucosamine is a natural component found in the cartilage. So they are present in the cartilage, glucosamine. N-acetylgalactosamine. It is necessary for the intercellular communication and it is concentrated in sensory nerve structures, both humans and animals. So they are also very, very important. So the last type of polysaccharide is the mucosubstances. They are slimy substances, mucilage-like, okay, and possesses acidic or aminated polysaccharides. So basically they are acidic in nature and they are mucilage. So wherever the mucilage structure is there, that is a type of polysaccharide, that is mucosubstances. They play important role in lubrication, protection, moistening and facilitating movement and trapping different substances even. Some of the examples of mucosubstances are agar. Agar, if you remember, we obtain agar from red algae. Example, Gelidium and Gracilaria. Pectin substances, hemicellulose and peptidoglycan. Some of the examples of the mucoproteins. So this is all about the carbohydrate. Thank you very much. We will meet again with one more interesting session of biomolecule. Thank you.